Hello, my name is Nathan and I'm a neuroscience PhD student at Columbia University. I'm here to introduce you to Late Night Science, a series intended to connect people and science together. We're going to be exploring the stories of people in science and the cool things that they get to do in their labs. If you like what you see, please join us in our in-person events in the New York City area. Details will be after the video. Tonight on this edition of Late Night Science, we'll be talking to Eliza Jaeger in Maria Tosche's lab. She'll be telling us about the evolution of the brain.
So my name is Eliza Jaeger. I'm a fourth-year PhD student in Maria Tosche's lab here in biological sciences. And I am generally just really interested in understanding how brains have evolved. This started out really early. When I was younger, my family moved around quite a bit, and one of the places we moved was Germany. We moved there when I was 11, and I had been there before, but I hadn't really learned the language. But then I was placed in a German school and allowed to kind of just absorb my language surroundings. And being 11 and still in that critical period of language learning, without formal instruction, I really just absorbed German and was able to speak it, and I still am fairly well. And so this really kind of kickstarted me thinking about, well, that is a pretty insane ability of the human brain to be able to simply be in an environment and listen and repeat and practice and learn an entirely new language from scratch. And it really just ignited my interest in the brain and specifically how we've evolved to process and understand language, and more broadly, just all of these interesting and complex abilities that the brain allows us to have. What looking at the amphibian brain tells us, it really tells us a lot of essential things about the building blocks that actually are important to make up vertebrate brains, right? Something that's actually stayed conserved across this many millions of years of time, that has to be absolutely essential in how the brain functions. So we work with salamanders because of their key position in the evolutionary tree. And we are some of the first people, one of the first labs to be able to try and leverage these animals for more systems neuroscience questions. We knew that part of our task in actually establishing these animals for understanding vertebrate brain evolution was to be able to build a single-cell RNA sequencing atlas of the different neuron types that are in the amphibian telencephalon or forebrain. So basically, when we do these kinds of analyses, you take a brain, and instead of being able to compare different structures or different parts of the brain between an amphibian and a mouse, which is a much more challenging thing to do, we basically take the brain apart into its composite parts, which are just its neurons, and you can sequence the RNA that's contained in each individual cell. And then based on those data, you can put cells in clusters based on how similar they are. You can kind of put everything together and say, okay, well, these RNAs are very similar in these clusters, right? And so we think that these cells between amphibians and mammals are actually more related than these other cells. So by understanding how the different cell types that are in an amphibian brain versus a mammal brain or a reptile brain, how they relate to one another, we can gain a lot of insight about the kind of brains that actually existed 350 million years ago, which was when our last common ancestor between mammals and amphibians actually existed. And in doing so, we can actually understand what has remained conserved. So things that you find in an amphibian brain and a mammal brain now, those things have actually remained conserved over 350 million years of evolutionary time. And you can also understand, all of the, of course, wild novelties that give us all of the really interesting and complex abilities that mammals, including us, are able to do. And this is a huge kind of step forward for evolutionary neuroscience. And there's a huge effort underway to sequence, cells from animals that are even further back in the evolutionary tree. So, jawless vertebrates, things that are, 600, 700 million years divergent from us. What are the even more essential building blocks of vertebrate brains? By sampling more species, we're going to understand more deeply the different kinds of evolutionary conservation that's taking place and novelties in the mammal brain. And so I was really interested to, first of all, see, can salamanders learn stuff? And can we measure it in the lab? And so what I set out to do was to be able to use stimuli that I knew salamanders didn't like. So they really don't like bright light. They'll go away from it. And actually get them to learn that an odor that they previously didn't care about, didn't mean anything to them, actually predicts that this light is going to come on. And then they actually form a new memory about that odor. And I've been able to do that in the lab to teach salamanders that an odor that they didn't care about before actually now has valence. It actually means something to the animal. My background is in behavioral neuroscience. So I, as an undergrad, I looked at, rats navigating in different mazes, and I'd had a lot of, experience doing mouse work, understanding mouse behavior. And so from that perspective, you come with a lot of expectations for what animals will do when you put them in a behavioral task in a laboratory setting. And I quickly learned that that was possibly unrealistic, and that I really had to kind of think outside the box behaviorally in terms of what I could expect our salamanders to do in a lab setting. One of the fun and exciting but also terrifying things about starting to try new techniques in an animal that really no one's used before to answer these kinds of questions is that you really don't have tools that other people have developed before you that you can say, okay, well, I know that these different salamanders have neurons that light up when they're active, so now I can use them to answer the kinds of questions that I'm interested in. You have a salamander, and you actually need to develop the tools to be able to answer the questions that you're excited about. So that's been a lot of, in addition to the behavior, I've been developing ways to actually measure neural activity in salamanders that no one's really tried out before. But that was just, one of those things where you're looking at a new organism for a behavioral assay. Things come up that you just didn't expect, but instead of being stressed out by them, we have really been excited to understand it a lot more deeply.
Thanks for watching this edition of Late Night Science. Make sure to tune in for our next speaker.